All right, so today, as Gary talked about, there's a lot of things that's been happening, and we've all had the opportunity to sit back and watch the news, read in print, read on the computer, on our phone, everything that's happening extremely fast in the human domain, and some of those technologies are very exciting, and I want to talk about some of those technologies as they exist today and what's uh, being brought forward into the realm of poultry vaccines. So these are the topics of discussion I will take you through today. So let's start with the introduction. Let's talk about history of vaccines, the different vaccine generations that exist, and of course what COVID-19 has done to these different generations. Specifically, we're going to talk about structural protein engineering. That's a tongue twister for me sometimes, and it's quite detailed, but we're just going to highlight that aspect because if it wasn't for structural protein engineering, we wouldn't have the aspect of these COVID vaccines that are working very well in the human population. As is the issue with adjuvant carriers and LNPs, the same goes with that. And then talk a little bit about synthetic biology, which is the basis of everything that we're seeing today. And then at the very last, talk about how this all ties into poultry. Talk about our existing recombinants for poultry and how we're advancing on that. The new vectors that are coming aboard, and of course the non-replicating vectors, which is basically circulated around mRNA or RNA, self-replicating RNA. So this image is basically brings back, how the population used to vaccinate against smallpox. This all started around the 10th or 11th century in China. For the most part, people would stand in line and have scabs of smallpox blown up into their nose to prevent, the worst part of the disease. This insufflation was something that was routine all the way up until Edward Jenner was able to come about and utilize a different virus for protection against smallpox. And modern history with Dr. Edward Jenner says, okay, listen, we're able to actually utilize the word vaccinate rather than insufflation to prevent disease in humans. And as that went further, of course, Edward Jenner is, most notably is where vaccination and vaccine discovery began. But Louis Pasteur was very active in this role too as well, with accidental serendipitous discovery of fowl cholera and into other diseases such as rabies and so forth. There was tons of activity in his laboratory, and including at that time as well, Ajeski and Joseph Merrick, which we all should know and recognize within the poultry industry. These advents and these discoveries in the 18th century, 19th century, early 19th century were tremendous for human as well as animal vaccination. And then as all this was coming to light, the United States government comes to be with the 1913 Virus Serum Toxin Act, which is basically the basis of our Code of Federal Regulations for how we produce vaccines and how we get vaccines out into the market, which are acceptable for the industry and safe for the industry as well as not only the animals but also humans. And soon thereafter, after that Virus Serum Toxin Act, the University of California had the first product license for fowlpox, and soon thereafter, Newcastle, ILT, and bronchitis came to be as licensed products as well. And then after the war, World War II, that's when the numerous vaccine companies came to light and started to operate inside the United States in producing, for the most part, kill vaccines as well as live attenuated vaccines. So therefore, what had happened is that we ended up with vaccines in several generations. And for the most part, if you look into your upper left of your screen, it all started with whole microbial microorganisms. This comes from Louis Pasteur, and even today we still utilize whole microbial microorganisms. We either grow them up and we kill them to make them safe, and we adjuvant them to make them safe when we put them in the host, or we grow them and we pass them in a medium that makes them weaker, makes them safer to give to the host. This is called attenuation. And this is still utilized today, but is becoming a methodology that is becoming less and less utilized as we move to different generations. Vectors or subunits, we should all be familiar with that because, for the most part, in the poultry industry, the recombinant Merix vaccines are quite readily utilized and utilized very successfully for not only against the fight against Merix disease, but other diseases such as Newcastle, IBD, and ILT as well. So, been very successful in the second generation as using vectors, and not so much subunits in our realm. Subunits have been more successful in the human aspect, but for the most part, we've been very successful in the use of vectors. The third generation, nucleic acids, has come to light recently with COVID-19, and this is something that we'll spend a little bit of time about talking today about how nucleic acids are being utilized not only in the human domain, but how they might be integrated into poultry, into other animal vaccines. So for the most part, what you're seeing today from both Pfizer as well as Moderna is the switch over to mRNA vaccines from the potential live vaccination. mRNAs are a little bit more advanced than your typical kill vaccines, and they are able to activate different arms of the immune system that historically kill vaccines have not been able to do. So therefore, the success that you're seeing or hearing about from these mRNA vaccines are real. And a lot of what you don't see is a lot of research and a lot of background work that has been done to make sure that these mRNA vaccines are working very well. And we'll cover a little bit of that as well. So basically, as it looks today, we have Pfizer, Moderna, and AstraZeneca leading the pack for COVID vaccines within the human health. Both Pfizer and Moderna are mRNA products, claims of efficacy, and of course, you've heard about their storage issues as well, but those have been addressed as more and more updated versions are coming aboard. Required doses are 2. Make sure you get that healthy immune response, and then side effects have been minimal, although you have been reading about several issues that are occurring in different populations. Cost per dose is pretty impressive for both the mRNAs, and of course more minimal for AstraZeneca, since it is a viral vector. But these costs that you see at the bottom of the page underneath the column for Pfizer and Moderna are actually coming down and will be coming down in the human realm. And this is something that is already being addressed in the animal health industry as well, as far as reducing the cost where they're applicable for both poultry as well as other livestock. So let's talk a little bit more about these particular technologies that have enabled the success of COVID-19 vaccines, as well will enhance the success of poultry vaccines as we move forward. First thing I want to talk about and spend just a little bit of time here is talking about structural protein engineering. This is basically the heartbeat of the success of the mRNA COVID-19 vaccine. Without this, the mRNA vaccines would not have been as successful as they are. This is very important to understand because this is something that is going to help the animal health industry tremendously as we move forward into developing our existing recombinant vectors, as well as our future products. Because what has happened is the ability to look at these proteins as they're presenting themselves to the host and making sure that they're presented properly. And what you see on the right-hand side is pictures of 2 different types of protein formulations for the spike protein for COVID-19. The one on the left is the preferred formation, prefusion spike, and as you can see at the top, there's more range for presentation of the epitope to the host versus the one on the right, which is the post-fusion spike, which is the non-preferred formation. And the issue here was the ability to produce this preferred version, the prefusion spike, to make it sure that it was stable once it's presented by the vector to the host, because this would quickly and rapidly go to the post-fusion spike version. And so this discovery happened around in 2015 and 2017, where they were able to implement changes to stabilize preferred prefusion spike. If you look at the bottom left of your screen, you can see the 4 or 5 spaces that are marked in red that indicate these small changes that enable this protein to stay in this prefusion spike presentation. So without this, the success that we see, no matter what the vector, would not be there without this type of discovery and without the use of the insert of the sequence needed to present this prefusion spike, which is stable and presents to the host, is able to elicit the proper immune response for protection against COVID-19. And therefore, you see the success not only against strains that are very similar, but also the existing variants that are popping up throughout the world, whether it's in UK, South Africa, or other parts of the world, that this vaccine has been successful in neutralizing these particular new variant viruses. So some very good news and some very good aspects in use of technology that can be transferred over to the poultry realm as well as other livestock. Another thing that's been highly touted and utilized is the use of lipid nanoparticles. These are, I guess, if you will, next generation adjuvants. Not really adjuvants, but they're actually carriers of this nucleic acid. And they allow for the entry of the nucleic acid into the host cells to let the mRNA work, or the self-replicating RNA work. But there's variable formulations that are utilized. This has been utilized more and more. They used to be very cost prohibitive, but now they are not, because the ingredients to make these become more and more common. And it allows for antigen optimization. So in other words, you don't need a whole boatload of antigen to load up into the host. You can minimize the amount of antigen you utilize, and these carriers are very efficient in delivering their package and presenting to the host. And here's the best part about it. You can now balance safety and efficacy. So the amount of adjuvant that you use here, the amount of lipid nanoparticles that you use here is very minimal. And so therefore the safety becomes a very easy to handle, and the efficacy becomes enhanced. So now, as we look into these future products, we see safety and efficacy becoming very balanced as we move forward in protection against disease. Very important aspect with the LNPs. So what this looks like in a production scale, and most of you might not be interested in production scale, but I think this is an important slide to understand, because this is how easy it is and how it can be readily utilized for vaccine manufacturers. Starting in your upper left-hand side, the target pathogen. For the most part now, we don't have to isolate the pathogen. All we need to do is have the nucleic acid sequences. And that goes to the second part, where we have the genomic sequencing. That analysis is very important. And as I mentioned, the ability to introduce changes that make sure that whatever you're trying to express, that protein will be very stable. And so that goes into vaccine design. And once that vaccine design is completed, then that moves more into formulation and production. And that is relatively easy today to create pilot mRNA vaccines that are mixed with LNPs. And these LNPs they can be assembled just as a very easy mix and go technology today. And it actually has reduced the temperatures that they're stored at. Ability has also been enhanced, and you can move to vaccination. So this is something that not only is fast and quick, but it also reduces the cost of goods that vaccine manufacturers often face and need to put in place in order to make sure that the final product is not cost prohibitive for the customer. So if we look at future vaccines, this is an interesting aspect, is that I think the future vaccines for the poultry industry have been here, and case in point is that we have one of the best recombinants out there in the term of Marek's disease, and the ability to use HVT as a vector is fascinating, and it is great across the industry, across multiple vaccine manufacturers. So it came with the advent of utilizing single gene inserts that would protect against a secondary disease such as Newcastle disease. So we would get protection with Marek's in Newcastle, and then we would also look at Vectormune IBD, also the same thing, against protection against Marek's and IBD. And then we would move on further to see whether or not these vectors will work together. Of course, what we found is that they're in competition with each other. So we had to take a step back to make sure that we could design a vector system now with two inserts that could protect equally across both IBD and Newcastle disease. And products are now coming out to where we have two genes of interest inserted into an HPT vector that protect against 3 diseases. So a fantastic movement for this particular vector, which is very good. It's the same vector that we've been utilized, but now we're actually moving towards new recombinants that are more effective. So we also are expecting new vectors to come out soon within the poultry industry, and this would be the basis of the synthetic biology that's being utilized today that was exhibited in the production cycle that I showed earlier. It goes as simple as on the left-hand side, top left-hand side, is submitting your sequence. You go through some complexity and biosecurity check, and you synthesize your gene. You can clone into the vector, and then you can make sure that it is confirmed by sequencing. And then you have the final product that you can deliver. This today can produce new vectors and new recombinants in less than 4 months, and can be utilized to rapidly assess the ability for the recovered virus to elicit protection against select diseases. So this type of technology has really moved forward in research and development, and is actually utilized today by several companies within it. I think what's most important too as well is to understand that our federal government has been very proactive in looking forward as far as what are we utilizing for protection against our animals in this field. It all started with the memo 800-213, where they allow for platform-based products, and these are products that were either vector expression systems. They allowed the replacement of gene of interest, and they must not produce infectious agents. So they're safe, but they allow for the rapid change of these particular products to fit industry's needs whenever new diseases are presented. And from this USDA memo, a couple of products have already come out into industry and are being utilized successfully within the swine industry, and have been utilized for stockpile purposes for certain diseases such as avian. Now, these technologies include virus-like particles, DNA plasmid vaccines, self-replicating RNA, and DNA and RNA, which is also been utilized in these particular non-replicated vectors, and include the technologies that we just talked about. Lipid nanoparticles are better adjuvants for the better carriers, nucleic acid, rapid assessment by genomic activity, and ability to put these together in a rapid format. But what we see across the world is that other people are following USDA's lead in this, and that we soon will have by either European Pharmacopoeia or by other countries the assessment of this particular memo, how it's being utilized, and implementation globally. So the take-home messages that I would like for you to have as we go forward, as we listen to these other talks about technology that's integrated into what we do on a daily basis. COVID-19 undoubtedly is driving technology changes for both human and animal health vaccine development. A lot of this you might not see right now, but you will see in the future. It's pulling into a technology that is safer and actually more efficacious than second and third generation vaccines that exist today. This rapid vaccine development is based upon virus sequences, and it becomes a reality. It becomes something that is being utilized today for research and development for future products, and will be soon something that will be that we will be looking at on a daily basis and be more attuned to as we go further. And what's important to understand is that these traditional live attenuated vaccines, which has been the cornerstone for animal health, as well as some human products, will enter into the history books. I think this has already started in human health, where we actually have seen a couple of live products actually being replaced either by subunit or by genomic vaccines. And I think you will see that very soon within the animal health industry, poultry industry as well, within the next couple of years. So these are take-home messages that I'd like for you to understand as we move in and as you recognize the technologies that are utilizing for COVID-19 that will soon be implemented in animal health as well as poultry health. With that, I thank you, and I hand this back over